When you think about all the life on Earth, I mean, all of it, from the tiniest microbe to the biggest whale, the sheer variety is just mind-boggling. So let's take a look at the massive biological filing cabinet scientists use to try and make sense of it all. Right? It's a huge puzzle, one that scientists have been chipping away at for centuries. How in the world do you create a system that organizes everything in a way that actually makes sense? A way that shows how all these different life forms are connected? For a really long time, the answers seemed pretty simple. The first big scientific attempt basically tried to split all of life into just two giant buckets. And this whole idea of sorting life isn't new. Aristotle took a crack at it way back when, using really simple visual cues. But it was Carl Linnaeus in the 1700s who really formalized it with his two-kingdom system. And that framework, it shaped biology for 2 whole centuries. The system was actually pretty elegant in its simplicity. Basically, if it had a cell wall and didn't really move, it was a plant. If it moved around and had to eat stuff, it was an animal. And for a while, that was good enough. It was straightforward, and it seemed to cover everything you could see with your own eyes. But as our tools got better, as we invented more powerful microscopes, scientists started finding these organisms that they broke the rules. They were like these awkward puzzle pieces that just wouldn't fit, no matter which way you turn them. And this gets to the heart of the problem. That simple two-kingdom system was really starting to show its cracks. It was becoming painfully obvious that this neat and tidy classification was, well, just not cutting it anymore. Okay, so let's break down exactly what was wrong. First off, it ignored these massive fundamental differences between cells. It lumped single-celled organisms in with complex multicellular ones. And maybe the biggest problem, it totally glossed over the huge distinction between simple prokaryotic cells, like bacteria, and the more complex eukaryotic cells that make up things like us. Take fungi, for example. We see these mushrooms here. Yeah, they have cell walls and they don't move, but they don't make their own food through photosynthesis. They're decomposers, so shoving them into the same group as green plants just because of a cell wall, it just felt wrong. And this was an even bigger headache. Bacteria are prokaryotes. They don't have a proper nucleus or any of the complex machinery that plant and animal cells do. Forcing them into the plant kingdom was like trying to hammer a square peg into a round hole. It was clear that something had to change. And then the solution arrived. In 1969, a biologist named R.H. Whittaker came along and proposed something revolutionary, a new system with not 2, but 5 totally distinct kingdoms. And this wasn't just about adding more boxes to put things in. Whittaker argued that we needed to look deeper, to use more fundamental characteristics to sort out the tree of life. We basically needed a whole new set of rules for the game. And here are his brilliant new criteria. Just look at the columns. He started asking better questions. First, cell type. Are we dealing with a simple prokaryotes or a complex eukaryote? Then, cell wall. Is there one, and if so, what's it actually made of? And finally, mode of nutrition. Does it make its own food or does it eat other things? These questions created a system that was so much more logical and scientifically solid. So with this new 5-kingdom map in hand, let's take a quick tour and see how it fixed all those old problems and gave us a much clearer picture of the living world. First up, Kingdom Monera. This was a genius move. Whittaker created an entire kingdom just for the prokaryotes. Finally, bacteria and all their relatives had their own home, a category that actually recognized their unique and simple cell structure. Next, he came up with Protista. This was the perfect solution for all those single-celled organisms that had complex eukaryotic cells, but weren't really plants, animals, or fungi. It kind of became this fascinating catch-all drawer for microscopic life like amoebas and algae. And you can just see the diversity here. You've got a dinoflagellate, which is a type of algae that can photosynthesize, and then you have a paramecium, which actively hunts down its food. In the old system, they'd be in totally separate kingdoms, but now they're both recognized for what they are, single-celled eukaryotes. It makes so much more sense. And remember our fungi problem? Well, Whittaker gave them their very own kingdom fungi. And the key reason is this word right here, chitin. It turns out their cell walls aren't made of cellulose like plants. They're made of the same tough stuff you find in the exoskeletons of bugs and crabs. That biochemical difference was the nail in the coffin. They were definitely not plants. And this brings us to a really crucial point. These classification systems aren't carved in stone. They're scientific models. And like all good models, they have to evolve. As we learn more, as we discover new species and develop incredible tools like DNA sequencing, our map of the tree of life gets more detailed, and our filing system has to change to keep up. Whittaker's 5 kingdoms brought so much clarity. But of course, science never stops. Today, a lot of scientists use an even more detailed 3-domain system that splits Whittaker's Monera kingdom into 2 huge groups based on deep genetic differences. It just goes to show you that organizing the story of life is a journey that's still happening. It makes you wonder, what new discoveries are just around the corner that will reshape our understanding all over again?